related to the anthocyanin less pomegranate, a unique composition of Pomewhite. So, first of all, let me introduce you with this AL pomegranate known as anthocyanin less pomegranate. Now, what is this word anthocyanin is? Anthocyanin is basically a pigment present into the fruit of the pomegranate and many other plants as well. But obviously, we are focusing here on the pomegranate one. So, the red color of the pomegranate and some more other color like pinkish tinge color or a dark red color or bright red color, all the shades of the red color, they are basically imparted into the pomegranate by anthocyanin. These are the pigment of the pomegranate. But the pomegranate that we are using with the green acre, this is basically the pomegranate which is anthocyanin less. This means that the pigment of anthocyanin is absent from this pomegranate. Now, how this is absent? Basically, in the plants, there are several mutations present which are beneficial to mankind. One such mutation is this anthocyanin less pomegranate. The wild type of pomegranate is red in color, while its mutated variants are present in so many colors. And among these variants, the most important variant is the variant in which anthocyanin is completely minus. Basically, how this happened to the plant whenever there is the synthesis of some pigment, there is a molecular biology level pathway that is running into the plant basically during the development process of the plant when the fruit is going to develop. At the time, the anthocyanin biosynthesis pathway that is controlled by a very important enzyme known as anthocyanin synthase. Uh, Due to the inhibition of this enzyme or due to the malfunctioning of this enzyme, the process stop here at the level of proanthocyanin levels. Basically, the proanthocyanins are the pigment like compounds that are not mature compounds. Now basically, this pathway is inhibited in the white pomegranate, which is anthocyanin less pomegranate. And as this pathway is inhibited, it will trigger some more important pathway to get initiated instead of the anthocyanin biosynthesis pathway. Now, the other pathways that are not triggered into this pomegranate, they are very important. They have very important medical and health benefits for human skin and some other organs as well. Here we are focusing on its benefits on skin and intestine and stuff like that. So basically, due to the reason of inhibition of this anthocyanin synthesis, the things that are going to increase in the anthocyanin less pomegranate, they include certain very beneficial phytochemicals and the derivatives of certain phytochemicals, because the final version of the phytochemicals are good for the skin, but their derivatives are best for the skin. And these are the natural derivatives that are present in the anthocyanin less pomegranate. Similarly, some vitamin content, and the most important is the prebiotic content. The prebiotic content, which is basically a sugar like carbohydrate, mainly the fiber content, this content is very, very high in anthocyanin less pomegranate as compared to the red variety. As when we eat the red variety and the white, we can very easily tasted that the red one is not very sugary in its state, while the white pomegranate is very sugary in its taste. So this means that it has a high content of the sugar in the form of carbohydrates, in the form of fiber as well. Similarly, due to the presence of these prebiotic in it also increases its content of antioxidants. Plus, it has the ability to trigger the antioxidant internal system of the cell of the body. We have checked it in vitro studies first in the lab with the help of cell lines, and also in vivo studies also, which is the clinical trial. So one by one, I will discuss these both of these trials. And now this anthocyanin less pomegranate, which is rich in prebiotic, it becomes the source of increase in probiotics. And as the probiotics increase with the representation processing, we will be able to clear more and more about the role of these two things in the acne and related things. All right. So the phytochemical compounds that are mainly present now with this anthocyanin less pomegranate are phenolic compounds and different type of flavonoids. Both are obviously phytochemicals, and within the phenolic compound we have allogytanins, allergic acid, gallic acids, and from the flavonoids, I have already told you that along with the normal flavonoids, uh, flavonoid derivatives are more present in this anthocyanin less pomegranate, because you see that these are also included in the category of the proanthocyanin pigments, and they are basically the intermediate components of the biosynthetic pathway of anthocyanin. That's why when the anthocyanin are not going to synthesize, then the intermediate of its pathway, they start increasing into the plant body. Are obviously they are very very beneficial for us, including the camphorol derivatives, quercetin derivatives, catechin and catechin derivatives. Um, Basically now this pomegranate become the source of attraction for many people because it is rich in prebiotic, high in the fiber content and sucrose. These are the main source of the prebiotic in it. And now what happened when somebody take the considerable on quantity of the prebiotic from this pomegranate, it will go to the bloodstream and then obviously go towards the skin layers. And here it causes the it causes to flourish the probiotic content of the skin. Uh, Basically the probiotics are the flora or the bacteria that are present onto our skin. They are basically the normal flora. Along with that, they are also present in the intestine. We all know that whenever we got some loose motion like problem or infection of the intestine, our physicians, uh, they advise us to take the diets that is rich in probiotics or prebiotic, because both of these things go side by side. Whether you take the probiotic directly, or you took the prebiotic, that ultimately will enhance the quantity of probiotic into the intestine. Similarly, from they are going to increase it in the intestine, and the same goes for the skin, because skin also is basically habitat for many bacteria, who are the who make the normal flora of the skin basically. But there are certain bacteria that are not the normal flora of the skin. They basically cause infection into the skin, and the most important thing is the acne related bacteria. And you know, this is a worldwide problem, especially in the young people. And the most important bacteria that is going to cause the acne, this is p acnes. The p acne is a bacteria with which can cause acne, and the probiotic have the ability to wipe it off from the skin. Similarly, this AL pomegranate also has some whitening benefits in it, because the balanced quantity of polyphenol like camphorol derivative, quercetin, catechin, and catechin derivatives, they all combinedly affect the skin whitening properties. And basically, they are doing it by decreasing the tyrosinase activity and inhibiting the tyrosinase activity. Now, once the tyrosinase activity is inhibited, the melanin synthesis also inhibited into the cell, and this is how the skin become whitened in its tone. Then very important other effect that is closely related to the whitening benefit is the antioxidant benefit. The antioxidants are already increased due to the presence of these uh, derivatives of the flavonoids into the AL pomegranate. But along with that, once this AL pomegranate constituent they get absorbed into the cell, they then trigger the cellular own machinery to produce antioxidant. This is how the double X antioxidant increase effect is there uh, from the normal medications with this AL pomegranate, and this is how the overall wellness of the cell will be there, and its antioxidant index become very high, and defense system become very strong. The key benefits that can be extracted from the Pomewhite anthocyanin less pomegranate include in vitro studies. They include the following steps. Like We have checked it on a different concentration onto the skin cells, and it is proved that on even at a higher concentration, it is non-toxic to the cell. We have done it with the help of cytotoxicity assays, and cytoprotective benefits or were also seen on Hackett cells. Hackett cells are basically the skin keratinocyte cell lines, and in vitro studies, we are using this cell, and these cells are responded the benefits of AL pomegranate extracts up to very high levels. I will show it in the detail in my results section now. Then this pomegranate is rich in prebiotic, and this prebiotic content, once they increase, they will obviously increase the gut microbiota, and then after the absorption they will increase the skin microbiota, and this is how the skin probiotics are increased. And due to the increase in the probiotics in the epidermis of the skin, it can prevent the problems of the skin like acne. Along with the acne, some other problems of the skin could also be diminished, like, for example, the control of the melanin synthesis by the inhibition of tyrosinase activity. Then they can increase the VEGF level onto the skin. These VEGF level are very important for the skin regeneration and blood circulation. And along with that, the inflammation reduction and the properties like this could be imparted onto the skin. This is a comparison. Here comes the result, and here is the comparative study of red and anthocyanin less pomegranate flavonoids content. HPLC chromatography was performed to do this. And here from the graph, you can very easily see that the red pomegranate has campherol derivatives in a much lesser amount than they are present in the AL pomegranate, and means white pomegranate. And the quercetin derivatives, they are also present in almost absent in the red pomegranate. If they are present in, they must be present in trace amount because they are not fall into the detectable range. So while in case of AL pomegranate, they are very high, and quercetin is a very very famous antioxidant and very famous flavonoid related to skin and other uh, organs of the body. Catechin derivatives are also present in lesser amount in red pomegranate, and they are relatively high in white pomegranate. While catechins complete mature compound, it is present in a very less amount in red pomegranate, and in white pomegranate it is high. So this shows very clearly that the derivatives are present in a high amount in AL pomegranate compared to the red ones, and this is the basically the beneficial effect that we can get from this red pomegranate along with its prebiotic content. Okay, so here comes the results of the cytoprotective effects. I have already told you that we use Hackett cells. Hackett cells are the skin keratinocytes basically, and they are in a culture form. A single layer of the cell is formed onto a plastic polystyrene surface, and then we apply different concentrations of this Pomewhite in onto these cells along with obviously their medium. Here we start from the 25 microgram per ml in a medium Pomewhite, and from the 25 microgram per ml till 3 mg per ml, we have tried it. This 3 mg means 3000 microgram. So after checking the absorbance, we come to the conclusion that the normal cell, who are the control cell, they have their proliferative index or their viability index up to this level. And after the treatment with Pomewhite on this Hackett cell, no cytotoxicity is seen. You can see it here that no cytotoxicity is seen. Instead of cytotoxicity, up from this 500 microgram level, we have almost a uniform conformation till 3 mg per ml for the Pomewhite. So we choose for further studies the two basically concentrations. One is this 500 microgram, and other one is the 1 mg, because the other assays that we have done on some other concentration, we got the best result with 500 microgram and 1 mg. So in vitro dosage of the Pomewhite are 500 microgram and 1 mg per ml, the best suited dosage for the cells. So here from this graph, it is now clear that this extract up to 300 mg per ml, it is completely non-cytotoxic, because if it is cytotoxic, then the viability of the cell graph, it goes down, like at this place or somewhere else. And if it goes down, this means the product is cytotoxic, and then we applied lesser dosage, or we recommend some changes in it. But obviously this is non-cytotoxic, so nothing is required, we can move it further as such. Then the most important thing is it can enhance the regeneration. We have checked the regeneration with the help of VEGF level at 500 microgram concentration, and at this 1000 microgram in 1 mg per ml concentration. At the both concentration, we can see that from the control, the regeneration ability is high in among this AL pomegranate treated keratinocytes. Another very important constituent of the epidermal and dermal layer of the cell are melanocytes. The obviously these are the keratinocytes, and keratinocytes do not have the ability to synthesize melanin in them. So for this purpose, we use another cell line known as A375. This A375 cell line is basically melanocyte cell line, and a very important cell line. The melanin is regularly synthesizing in these cells. We have applied this 500 microgram per ml dose and 1000 microgram per ml dose onto these melanocytes, and after 24 hours of the treatment, 48 hours of the treatment, we saw that uh, the level of the tyrosine, a tyrosinase enzyme, they become very decreased into this 500 microgram per ml dose, and they are even more decreased with 1000 microgram per ml dose. This shows that tyrosinase activity is inhibited, and once the tyrosinase activity is inhibited, this means that melanin synthesis biosynthesis is also inhibited. And when melanin biosynthesis become lowered into the skin looks very radiant, and the whitening properties imparted into the skin. Similarly, after doing this, we focused on the increase of probiotic content, because we have already checked it, the prebiotic content of the pomegranate with proximity analysis, and this proximate analysis, it shows that it is high in the prebiotic content. So just to check that is it is going to increase the probiotic or not, we applied these two concentration on a commensal of the skin known as Staphylococcus epidermidis, and uh, the skin acne causing bacteria, which is known as Propionibacterium acne, which is also known as P. acne. So this S. epidermidis and P. acne, these are the two candidates, the main candidates of the skin. This P. acne is high with the skin of acne causing acne skin people, while S. epidermidis is a normal inhabitant of the skin. We took the culture of these two in vitro, and we apply our these two concentration of AL pomegranate on them. And here we saw that in case of S. epidermidis, it is going to increase somehow from the treatment with the AL pomegranate, while a very significant decrease in the P. acne was seen with the help of this. Then the basic mode of action of the bacteria, these prebiotics and probiotics onto the acne bacteria, is this that whenever the probiotic content of the skin got increased, it basically released different type of proteinase contents onto the skin layer that can eradicate the acne or infection causing bacteria from them. So these are basically our in vitro trials, and after completing this trial, which shows that it can regenerate the skin, it can whiten the skin, it can combat the acne problems of the skin, we move towards the clinical study. From the in our clinical study, it's basically a double-blind, placebo-controlled trial, and here we have almost 100 volunteers with us. Basically, we recruited more than 100 volunteers, but 100 volunteers are chosen on the basis of their level of acne, that is identified by dermatological panel that moderate to severe acne people are recruited, and 20% are recruited who are healthy volunteers. Their skin type is considered as healthy because they do not have the acne problem, and their skin type is also normal skin. The duration for the study is 8 weeks, which is 56 days, and we have the sampling dates of at 0 week, means before the treatment. We have done the sampling of the blood and the swabbing and the serum, and after 4 weeks, again the same procedure was done, and after 8 weeks, again the same procedure was done. So basically, we have the three times the sampling from the participants. The test group are basically the people, all the people with moderate to severe acne. The dosage that we have applied are in the form of the capsules, and these capsules are 150 mg and 3 mg of Pomewhite that are given to the participants. Along with that, in the placebo group, we are giving them 150 mg and 300 mg capsule with rice powder, because the rice powder is sort of an inert material, and instead of Pomewhite, if for placebo group people, the capsules are filled with the rice powder in the same quantity. Primary objective of our clinical trial was to evaluate the Pomewhite for decreasing the amount of acne. Now, how it is going to decrease the amount of acne? Mainly by increasing the probiotic and control of the oil production of the skin. So these two are the key features of the Pomewhite. Along with that, some other factors are also present that further have their potential effects for decreasing the amount of acne from the skin. And these include a very important is the inflammation reduction. We all know that whenever there is acne on the skin, we can identify on acne skin very easily three, four problems. First one is obviously the acne lesions or pimples, they got infected very easily, and a big scar can leave after after even the antibiotic treatment. Second thing is the acne lesion. It got inflamed. Whenever there is a severe acne or even in moderate acne, there are certain lesions of the acne that become inflamed, and the areas around the lesion it become inflamed. So obviously there must be a therapy that can combat with these inflammation like problems. Similarly, another very important thing is that after the acne, the scar remains onto the skin, and due to the numerous scars present onto the acne person's skin, the skin become tanned, and the skin tone is going to disturb. But if there is some therapy that can has its effect along with the inflammation reduction for the scar reduction, obviously it will give the brightened look that can be achieved by the Pomewhite. Then after the acne lesions or the pus of the acne lesion become wipe away from the skin looks scary, because you see the scars present on the skin are there, and some fine lines also appeared, because whenever we got some medication for the acne, the allopathic medication basically, then all these products, the allopathic medicines, they are the main cause of excessive dryness to the skin, and the fine lines and small wrinkles they appear onto the surface of the skin. So basically, the premature aging of the skin, you can say, is there in the acne people. So the removal of these fine line is a unique ability of the Pomewhite. Similarly, it has also a very unique ability of antioxidant stress removal, and this removal obviously contribute to the overall wellness of the skin. The skin look radiant, and our defense system, antioxidant defense system, it becomes strong with the use of the Pomewhite. Here is the study design, and in the study design we have 20 healthy volunteers who serve as the normal group. Then rest of all the groups have the persons with mild to severe acne. But they are categorized as 20 volunteers are categorized in group 2, known as untreated group. Then the group 3 is the T placebo group, which is treatment with placebo. T plaque means treatment with placebo. and 150 mg per day. This means that the rice powder capsule 150 mg per day. Basically, the capsules formulation are 150 mg each capsule, whether it is a rice powder or it is Pomewhite. Both are 150 mg, but we designed the way like one capsule per day or two capsules per day for every volunteer. So the group 3 basically is T placebo group with 150 mg per day of the rice powder. Group 4 is T Pomewhite group, means treated Pomewhite group, and it is 150 mg per day Pomewhite capsule. Group 5 is the treatment group with T placebo 300 mg per day, means rice powder 300 mg per day. And group 5 is the last group, the treatment with Pomewhite 300 mg per day. This means that Pomewhite two capsules per day for the volunteers till 8 weeks. Here is our study design. First step is the recruitment of the volunteer according to criteria, and the major point of our criteria is that they do not use any antibiotic or drug since 2 to 3 months, and non-pregnant women are included. And obviously, with the men, this problem is not there. But if we include the women, we see this, and then they are not going to use any they just wash their face with regular face wash. No other medications should be applied during the course of this study, and they give us the consent, and then we start the study. We give them the capsules of placebo or Pomewhite, and after that their assessment of vital is done in at day 0, day 4, week 4, and week 8. This is mandatory to record their vitals include some of their LFTs and their blood parameters and their blood pressure, their SI index, which is shock index, uh, so stuff like that. So these vital assessment is very important because we can see from these three level of values at 0, 4, and 8 weeks that our study is our product is going to disturb the vital, or it is not going to interfere with the vitals of the um, volunteer. The main property then that we study, as we saw this property in our in vitro studies, so we are focusing it on in vivo studies as well. So this is the main focus is the level of acne measurement. This level of acne measurement is done by various tests. First is the skin microbiota assessment. We all know that whenever we got some infection of any part of the body, the doctors they recommend us to have the culture study of that tissue. The culture study means that microbiologists they identify different type of presence of bacteria, and then they identify that this much bacteria are pathogenic on our tissue, and then they recommend the medication. We here with our sample after the swabbing, we send these samples to microbiology lab, and then here we assess the presence of skin commensal bacteria and uh, acne causing bacteria, infection causing bacteria, both at 0 a week, at 4 week, and at 8 week, at three levels. Then the very important activity, which is the related to acne, is lipase activity, because in almost the acne problem is related to the oily skin people are prone to the acne problem. This could be one reason for the acne, that their lipase content is high, the lipase activity is high. Basically, this lipase is the enzyme that is present into our bloodstream, and it can break down the lipids are present in every cell. Lipids are present in every cell membrane, but the lipase, what it is going to do, it's just break down the lipids, and these broken lipids they then available onto the skin with the sebum secretion. And the sebum secretion, once it's increased, it will aids in acne formulations and acne proliferation. So we have measured the lipase activity at different time point at 0, 4, and 8 weeks. Then, with the help of questionnaire, we also calculate our visual analog, the level of satisfaction and stuff like that. And some morphology assessment, like the pictures of the volunteers are taken, and we quantified these photographs that whether the level of acne is going to reduce with our treatment or not. Along with this main measurements, we have the evaluate different other properties that include: number one is the assessment of skin aging. This is done with the help of measuring the fine line and wrinkle. This assay was done by expert dermatological panel. They people convert the fine line and wrinkle quantity into a numerical scale of 0 to 8, and we then calculate them from the numerical value. Then we have the assessment of skin inflammation. Two very, very important markers of skin inflammation are interferon gamma, which is an anti-inflammatory protein, and interleukin 4, which is an inflammatory protein. So these two are opposite to each other, and we assess that after the Pomewhite treatment, which is going to increase and which is going to decrease. We will see it in the result section. Similarly, the skin evaluation of skin stress was done with the help of two markers, in which one is SOD, which is superoxide dismutase. This is a very important enzyme, and this is antioxidant enzyme basically. And another very important antioxidant is glucose 6 phosphate dehydrogenase. This is very important enzyme because its substrate is glucose 6 phosphate, and glucose 6 phosphate comes directly from the carbohydrate content. That's why uh, this is measured in many, acne problems. Then we have the evaluation of skin brightening with the help of tyrosinase activity, because in our in vitro studies, we have already saw the melanin inhibition with the help of anti-tyrosinase activity on the A375 cells. So here we are using this activity as well. Now the treatment groups include: number one is the normal group, that is the person with skin type 1 and 2. Basically, these skin types are also designed for our ease, that skin type 1 and 2 almost fair complexion, and normal skin type, not very much oily, not very much dry. They are included in type 1 and 2. 20 volunteers respond up to 8 weeks. Basically, this is the number that we recruit in these different groups, and at 8 weeks, these are the numbers that respond us. Basically, in our T placebo group, the respondent number is 20. In all other groups, the respondent number is somewhat higher than the 20. But because we have this least number in our one group, that's why we make all our groups up to 20, and we calculate the results of these 20% from each group. With the untreated group, we have 3, 4, and 5 skin type. These are the types of the skin in which there are acne pimples and acne lesions present, scars are present. They have fine lines along with the side of their lips, and they people are having no treatment. From not placebo and not the Pomewhite. So they will become our untreated group, which is group 2. The third group is T placebo group, in which the acne skin people are there, the same group 3, 4, and 5, but they are with rice powder 150 mg per day treatment. Then we have T POMI 150 mg per day group. In this group, again, this 3, 4, and 5 type skin acne type are present, and they are treated with Pomewhite 150 mg per day. Then we have T placebo 300 mg per day, in which again the same type of skin are recruited, and they are treated with the rice powder 300 mg per day, means 2 capsules per day. While T Pomewhite 300 mg per day group constitute the sixth group. It has 3, 4, and 5 skin type acne people with the treatment of Pomewhite at the concentration of 300 mg per day. This means 2 capsules per day of Pomewhite. So the assessment, the first thing that now we have done from this sampling is our assessment of skin health and microbiota sampling. These are the primary objectives to evaluate the Pomewhite. For this purpose, the bacterial index was measured. Bacterial index measurement means the culture studies in which the probiotic or normal flora and acne producing bacteria colony, these two colonies are counted after 48 hours of culturing, and then acline count was there, that are further expressed as the level of acne by expert dermatologist. Then inflammation index total number of lay-ins were recorded at every follow-up. Then we have a very important property of antioxidant enzymes, and for this study, in order to measure the ROS, which is reactive oxygen species, we include the enzyme superoxide dismutase and glucose 6-phosphate dehydrogenase. Both of these are very important in the pathway of antioxidant defense system. These are the treatment groups. Sorry. Okay. So the estimation of superoxide dismutase and the estimation of glucose 6-phosphorydehydrogenase. These two estimations were done with the help of the kit and according to their manufacturer's protocol. And the results of the kit are expressed as their percentages and percentage activities, etc. Then a very important assay related to the proteins measurement is enzyme linked immunosorbent assay. From this assay, with the help of a previously reported Roger et al. Method, our IL-4 inflammation and interferon gamma inflammation, we check both of these markers with the help of our ELISA system. Um, Then the most important activity that we have measured with the help of kit is the estimation of lipase. Again, the lipase estimation kit was used, and the percentage of lipase activity was calculated according to manufacturer's protocol. Because this lipase is increased in the bloodstream of acne people. Then we have and tyrosinase activity measurement. Again, the percentage activity of the tyrosinase was measured. Tyrosinase is basically a copper containing enzyme, and it catalyzes the production of melanin. And if we some with the help of some way, if we could inhibit the tyrosinase, this means that we are going to inhibit the biosynthesis of the melanin and some other pigment. Because basically what happens is that tyrosine is a compound that is present in our bloodstream and in our skin and in obviously every organ. This tyrosine is oxidized by tyrosinase enzyme, and this oxidized tyrosine then is being used for the synthesis of melanin and some other pigment. So if we are going to design a strategy that can inhibit the tyrosinase, it will further inhibit the oxidation of tyrosine, which is not now available for the production of melanin. This is how the melanin synthesis is decreased in the skin with less and tyrosinase activity. And we have assessed this in our Pomewhite treatment groups. The trial schedule, if we could see that it lost almost for 8 weeks, and the food test product, they are supplied in a folded container, and the clinical assessments was done at day 0, 28th day, and which is 4 weeks, and 56th day, which is 8 weeks for all the participants. At every visit, we have collected their blood for serum analysis, and basically blood is taken in two type of tube. One is the EDTA containing tube, in which we got the blood with for glucose 6-phosphate dehydrogenase analysis, and in another container that contains some clotting agents in it with the gel tubes. We can also collect the blood in that tube. Here from this blood serum was isolated. This serum was then subjected to ELISA and certain other studies like inflammatory markers and superoxide dismutase analysis. Then from the cheeks and area around the lips and from the forehead, we have the swab cultures of them, and of different type of skins in different groups. Then we send these swabs to culture the bacteria, and from the bacteria we assess the level of presence of colonies with the commensal bacteria, and which is the normal flora of the skin, and the acne causing bacteria. And again, this test was done at day 0, week 4, and week 8. Skin tone was checked with different treatment group in post-treatment examination by dermatologist, and the number of inflamed and non-inflamed lesion was also recorded in order to check the level of acne. And then some of the parameters of level of acne were also calculated with the help of the different type of questionnaires. After doing all these analysis and this, we checked our data statistically as well. The software that is being used is the GraphPad Prism software, and in the GraphPad Prism software, all the experiment groups value they express as standard error and the mean, and the experiments were repeated in triplicate amount. For statistical triplicate, here the triplicate means that for every person we got the test three times. That three times we measure the person's SOD, three times we measure its uh, level with different ELISA and stuff like that. For statistical analysis, the group means they are compared by one-way ANOVA, and Benfroni's test it was used as post-test for comparison, and this all was done automatically with the help of the software. And the quantitative data basically obtained from different studies, different experimental group, it was statistically analyzed by using two-way ANOVA in GraphPad software, which is Prism 5 version. Then different type of signs are used in it. For example, static sign. This static sign in every graph, it will show the intra-group comparison. Intra-group means if we are having a group of week zero, so this means that different parameters of week zero, like normal, untreated group, group three, or group four comparison within the group zero, this will denote from static. And three steric mean high level of significance, two steric mean moderate level of significance, and one steric mean relatively low level of significance. Then we have intergroup comparison. This intergroup comparison will be denoted by alpha and beta. So this comparison is between different type of group. Like alpha, if we talk about alpha, so alpha symbolizes the comparison of 0 weeks with group 4, and basically with the group 4 and 8 of 150 mg per day treatment. While in case of beta, beta basically symbolizes the comparison of week 0 treatment group 300 mg per day with the both treatment groups of 3 mg per day, means week 0 and week 8. This is how we compare them. Sorry. These are the study results. Now, from the study results, it could be clearly seen these are the vital function parameters assessment. From the vital function parameters, we can see that all these vital function up to SI index. You see, this is the direct bilirubin. It falls under the category of normal range. Total bilirubin also is in the normal range. Even after 8 weeks of treatment, it is in the normal range. Similarly, alkaline phosphatase, it is again in the normal range. Vitamin D levels are also in the normal range. A very important thing that I want to discuss with all of you here is that whenever we have some clinical trial related to especially the oral use of any drug or any nutraceutical or anything, at that level, we have to have their liver function test to be normal, because their liver function tests are very important. We know that the liver is going to detoxify different type of drugs in our body. So if somebody is going to take some hazardous chemical, then the first thing that is going to disturb in his body is liver function. That's why with every clinical trial we check the LFT level, liver function test levels, and this level could be measured by these three major markers: total bilirubin, direct bilirubin, and alkaline phosphatase. Then vitamin D levels were also checked along these among these groups, and they also fall in the normal range. Random glucose levels are checked because we already know that uh, many people say that the Pomewhite is rich with the sugary content. So is it is going to increase our sugar level? No, this is not like that, because it's actually not actually the sugar. the good sugars in the form of fibers, in the form of sucrose, they are present into the palm white. That's why it is not going to disturb our glucose level. Here from the graph, we can saw this very easily that random glucose levels are fall under the. Then this is not fasting one, because if it is fasting, it should be almost 80 or 90. But because it's a random glucose level, they are also at their best place. Then the very important thing is SI index, known as shock index. The shock index is basically the ratio of our pulse rate with our systolic blood pressure. So, and this ratio should fall in a very tight range from zero point 0.5 to zero point 0.7. If your SI index is from zero point 0.5 to zero point 0.7, this means your heart is going well and it is doing perfectly fine. And if your SI index is more than zero point 0.7, obviously there is something to worry about now. So here the SI indexes also fall in the ideal levels during the course of study, means during the 8 weeks of the study. Here comes the real results of our study, the very important results, in which the first one by one I will discuss all the aspects. Number first is our primary objectives, known as skin tone, skin acne decrease from the palm white. Here, after the palm white treatment, now we all know that we have treated it with the palm white for at week 4 and at week 8 and at 0 week as well. So these three graphs are at week 0 graph, week 4, and week 8 graphs. In week 0 graph, we have normal person skin cultures, and then we have all our other skin cultures. Here, we can very easily see that in our all the untreated group, the level of normal probiotic flora, they are less than the acne causing bacteria flora, because obviously these people are with moderate to severe acne problems, and that's why their flora has more acne producing bacteria, while in case of normal people, very few bacterial colonies could be seen that could cause the infection of the skin. Otherwise, all their flora, almost 90% of their flora, is the normal flora of the skin. Then after week 4, obviously the normal skin has the same quantity of almost the same quantity of the flora. But in case of our treatment groups, we could see now that in the treatment group again, the flora of acne causing bacteria is much more. This is shown in black color, while the commensal bacteria or the normal flora is reduced. In case of placebo, no difference could be seen. But in case of Pomewhite treatment, a slight increase was there. Similarly, a slight more increase was there at the 300 mg dose, but after 4 weeks of treatment. As we keep on using the Pomewhite on the volunteers till 8 weeks, the 8 weeks show remarkable results, because now at the week 8, we saw that our acne producing flora is highly eradicated from the skin of the people in 8 week post treatment, 300 mg with Pomewhite, and their commensal bacteria or the probiotic bacteria, they are going to rise. These are the same things that we got in our in vitro study. In our in vitro study, the commensal bacteria, they become increased, and P. acne bacteria become decreased. In after the treatment with Pomewhite, same results are found from the clinical aspects, that again the acne producing bacteria, they become eradicated from the skin, while in case of this Pomewhite, we have got high number of commensal bacteria. Now the probiotic content of the skin being increased. Then the skin acne, for the skin acne decrease, we also study the lipase activity. I have already told you what is the function of the lipase, that it is the enzyme that is going to break down lipids in our skin, and due to the presence of these lipids onto the skin, more and more sebum secretion is there. This sebum secretion further aid in the proliferation of the bacterial cultures of P. acne onto the skin. Now, after the Pomewhite treatment, if we see the lipase activity, its percentage analysis, we can see that in case of normal persons, a very less amount of lipase is present in their serum of the these normal persons, while in case of acne persons, this level is very high. After the treatment with Pomewhite, it start decreases with the passage of time. And you can see that after 8 week of treatment with 300 mg per day treatment, we have a highly significant reduction in lipase activity. This is how we said that the inhibition of lipase activity is there with the tea palm white, and this is almost a 50% reduction. And obviously, when this sebum quantity is controlled and oil quantity is controlled onto the skin, this will impart the decreasing of the acne as well. After carefully examination of the questionnaires, the visual analog was calculated, and this visual analog also is calculated on a numerical scale value of 0 to 10. And from this calculation, we can see that in the normal persons, the visual analog of the skin is very high, a very radiant skin is there with the normal person, the skin type 1 and 2, while in case of acne person, along with multiple skin problems, obviously its level is decreased. After the treatment with the palm white, their levels are again increasing day by day. And at 8 week of treatment, we got a considerable increase, obviously not equal to normal, but moving towards the normal levels with the visual analog. Similarly, the percentage analysis of visual analog also prove this fact that almost 2 to 3 times of increase was there in case of visual analog. This means that the wellness of skin increases in the acne people so much in after the treatment with palm white. This is the very important essay of our study, because after the visual analog analysis, we got the pictures from DSLR camera of different volunteers before treatment and after treatment. Now, this before treatment or after treatment photographs were then analyzed with the help of image software. This is a software in which we can break the pictures into three different filters, and with the help of these filters, we one can calculate the redness on the skin. This red color is basically imparted the portion of the skin with relatively reddish tone, or the marks that are red in tone. You know, the scar of the skin due to acne, and the pimples of the acne and acne lesions, all they are reddish in their tinge. So this means that all the area of the red color on the skin in the pre-treatment groups where there is no treatment with palm white, it is more. While at the same level, when we check the other figure, when the person is completed the treatment of 8 weeks, the scars and the marks they reduce very much. The same could be seen from naked eye, and after the careful calculation, we got these results. Similarly, another volunteer with heavy scars on the skin, and after the treatment, 8 weeks, she also has very less amount of scars and pimples on her skin now. So this is the very astonishing results. Uh, then very important thing is the level of acne, that is again defined by a dermatological panel at a scale of at a numerical scale of 0 to 5. Here from the level of acne, it can be seen that the level of acne was high in case of untreated or the persons with acne, while in case of normal persons, level of acne was obviously very low. And then as the palm white treatments move on increasing, we can see that after the 4 weeks of the treatment with 150 mg, no significant changes was there. That's why uh, we haven't put a significant sign over there, but 300 mg dose have some significant changes in even at after 4 weeks. But after 8 weeks, we got astonishing results with the uh, tea palm white at 300 mg per day levels. Their levels once calculated with the percentage calculation here again, almost 40% of the decrease in the level of acne could be seen after the treatment with palm white for 8 weeks at a concentration of 300 mg per day. Then the other properties, which are the secondary characteristics of the palm white, but obviously very, very important secondary characters. They include number one is superoxide dismutase level. These levels are again very important in our antioxidant defense system. Whenever there is increase in the acne in the bloodstream, SOD levels are going to be high because of the presence of the reactive oxygen species. When the people they are treated with the Pomewhite, their salt levels are decreased again with 150 mg per day levels for 4 weeks and 8 weeks. No significant change could be seen from SOD decrease, but with the 300 mg level, high significantly decreased of the SOD was there. This means this group is best suited for combating the reactive oxygen species into the body. And obviously, when reactive oxygen species is minimized into the body, it will contribute to the overall healthiness of the skin and wellness of the skin. Here, the percentage analysis could be seen, and 25% decrease in SOD levels is reported. Then we have another very important marker is glucose 6-phosphate dehydrogenase. And this basically a enzyme that can have the dehydrogen properties in the glucose 6-phosphate, which is a substrate for this enzyme. And this enzyme is increase in normal person and decrease in the acne related persons. And this level is found to be increased in 300 mg per day Pomewhite treatment with the high significant levels, while in case of 300 mg at week 4, low level of significance was there. But in case of 150 mg, almost negligible increase was there. That's why no significant levels could be seen. The percentage analysis of glucose 6-phosphate dehydrogenase also show the same results. Almost 3 times increase of this enzyme levels was there into the Pomewhite. Basically, the enzyme known as this glycerol 6 phosphate dehydrogenase. This is very important when we talk about the prebiotic digestion, and the effective digestion of prebiotic then obviously aid in the high increase of probiotic contents. Then we have the tyrosinase activity analysis. This tyrosinase activity analysis was also done from the serum of the blood, and from the serum it is seen that their activity is maximum in the acne people. And as they people are using Pomewhite day by day, their levels are decreasing, and at a level of almost 300 mg per day, their level is decreased so much that almost 40 to 45% decrease was there in case of 300 mg per day Pomewhite treatment till 8 weeks. This will impart the skin whitening properties to the skin. Then the fine line and wrinkles are also reduced along with the scar reduction and along with the whitening properties and antioxidant increase. The fine line and wrinkles are decreased, and within the numerical scale of 0 to 8, we can see that in the Pomewhite treatment group 300 mg per day till 8 weeks, we have got a decrease in the fine line and wrinkle. Their percentage analysis also revealed the same thing, almost 45 to 50% decrease was there uh, in the fine lines and wrinkles. Then the very important property of Pomewhite is that it can reduce the skin inflammation, because this inflammation is a hallmark for this the presence of the scar and acne lesions. And once the inflammation is diminished, then the acne lesion size become very much reduced. So in case of uh, IL-4, which is a very important interleukin, this is a cytokine interleukin, and it is going to increase in the inflammation. This is basically a pro-inflammatory cytokine that in the start of inflammation, it start increasing in its quantity. And from the graph, it is shown that the IL-4 levels are decreased in the Pomewhite treated group till 4 weeks, and their level are decreased up to moderately significant levels. And the same goes for the percentage analysis curve. Here again, almost the 50% level of inhibition of this IL-4 was there. Similarly, another important marker for skin inflammation is another cytokine known as interferon gamma. This interferon gamma is a anti-inflammatory cytokine. Once the interferon gamma is released into the skin, it will cause the decrease in inflammation onto the skin and other body parts. Uh, in the normal persons, a considerable amount of interferon gamma are always present in the skin, but with the inflammation of the facial skin or other skin parts with some inflamed lesions, the levels are become low for interferon gamma, and once the volunteers are treated with uh, T-POM white up to 300 mg per day, they basically then have their interferon gamma level again revert back towards the normal one. And as this level is high, this means that the number of inflammation around the lesions they become very low. Here from the percentage analysis again, this is revealed that almost three times the level are increased in case of treatment with 300 mg per day palm white. So, based upon all these findings that we have with our clinical trials, we can conclude very easily now that the unique formula of palm white augment the increase of probiotic level in acne damaged skin, and this will definitely not only eliminate the acne, but it will also contribute to repair the damaged skin, and regenerate the skin. It enhances the skin tone with the help of this anti-tyrosinase activity. Then it also controls the oil production of the skin by minimizing the lipase activity. Then it also combat with the reactive oxygen species and inflammation by different markers, as we have already discussed. So this means that palm white could be a potential viable option for the treatment of acne damage and inflammation and improve our skin health. The safety study for in vitro studies is from 25 microgram per ml to 3000 microgram per ml, which is 3 mg per ml. These are proved by with the help of cytotoxicity done by MTT assay protocol. Then target indication, if we talk about the target indication, then we can treat a different type of skin problem with the palm white, like we can treat it with acne prone skin problems, then hyper pigmented skin and the aging skin, and we will be having good results with all these type of skin types if they are going to treat with this palm white. And the recommended dose for palm white are 150 mg to 300 mg, because you can see from the results that even at 150 mg dosage till 8 weeks, we got considerable results, but with the 300 mg per day, we have got remarkable results, but the range is there for 150 to 300. There are different suggested formulas as well, with the oral and the topical use of this palm white. On these formulas, right now the lab is going to work with, and obviously they will be in the market soon. Thank you so much. This is all from my side. And now, if you have any questions.